Okay, so this is going to be hopefully a quick lecture on meiosis. For your reference, you can look at your textbook. It's sections, I think, 14.12 to 14.14. So this kind of coincides with that material. Let me go to the slideshow real quick. We're covering the last couple of objectives for Module 5, which is a comparison between mitosis and meiosis. So we'll look at some of the differences. And then also looking at gametogenesis. How do we create these highly diverse gametes through the process of meiosis? For this lecture, we'll start with the mitosis-meiosis comparison, and then we'll look at some specific types of meiosis. We typically go through the mammalian meiotic cycle, or reproductive life cycle. But there are other types of meiosis that you should be aware of that occur in either plants or protists or fungi. So we'll talk about the three main types. And then we'll quickly review the phases of meiosis. This is where I think you need to make sure that you read the textbook and kind of review these on your own. I do have the accompanying worksheet for this, and make sure that you go through the worksheet that's posted on Canvas over meiosis to make sure that you are clear on what is happening at each of the stages of meiosis. Then we'll talk about how do we generate genetic diversity during meiosis. So what are the specific processes that allow you to generate that diversity? And finally, go over the steps of gametogenesis and what can happen if those processes go wrong. Okay, so very, very basic comparison, contrasting mitosis and meiosis. Hopefully you can fill these blanks in. So if we look at mitosis, what type of cell produces what type of cell? We're talking about somatic cells. This occurs in somatic cells. Meiosis will produce haploid gametes in most cases, and so this is taking place in the germline cells and will be creating gametes. So let me write that up quick. Cell produces for mitosis. Oh no. Oh, my markers are good. Okay. Oh, this is terrible. Mitosis is somatic cells. Let's try green one. Meiosis. This is producing gametes, haploid gametes. This is taking place in somatic cells. Meiosis is taking place in germline cells. And produces gametes. We could also call them sex cells. Okay, so hopefully you can see that. The amount of DNA that is produced, so mitosis, when we have a mitotic division, the daughter cells have the same amount of DNA. So you go from a diploid cell to another diploid cell. And in meiosis, you're going to go from a diploid cell to a haploid cell. So the daughter cells have half the amount of DNA in meiosis. So the DNA content here, so this is looking at DNA. This is the same amount versus half the amount. And this is basically going 2N to 2N, 2N to 1N, haploid amount. The identity of the daughter cells in mitosis, you're producing genetically identical daughter cells, or you should be. And in meiosis, we're creating genetically diverse cells. These are going to be cells that have a variety of different alleles, right? Okay, so how does meiosis work? Meiosis, unlike mitosis, which was a single division, is actually two divisions. Sorry, my computer's dying. It consists of meiosis 1, 2 rounds of division. Meiosis 1, which is called a reductional division, where you are having the number of chromosomes. So if you start with 2N, by the end of meiosis 1, you have 1N chromosomes. Meiosis 2 is a second division that follows. And in this division, we consider it an equational division because we do not have a change in the number of chromosomes. So it begins 1N and it stays 1N. So just to kind of demonstrate this here, these cells at the end of meiosis 1, you can see that in the beginning, we had 2 chromosomes in the cell, and by the end of meiosis 1, there's only 1 chromosome. Kind of reviewing, let me take the pen here. At the beginning of meiosis 1, we have 2 chromosomes. Okay, I can hardly write this. 2 chromosomes. And in this case, we had 4 chromatids, right? I'm just going to write tids because that is shorter. So 4 chromatids. By the end of meiosis 1, we only have 1 chromosome, but we do have 2 chromatids, right? So the sisters still have their pairs. But our ploidy number changed from the beginning to the end of meiosis 1. Now when we look at meiosis 2, going from here to here, you can see that the number of chromosomes is still 1. Still 1 chromosome. But by the end here, we only have 1 chromatid. 1 chromatid. So this is called a equational division, and the ploidy stays the same. Okay, so let's look at the mechanics of how this actually works. Well, actually, before we do that, quickly go over the different types. We typically, in most of our classes, teach about gametic meiosis, and that is the form of meiosis that occurs in mammals. It's also called terminal meiosis because basically the meiotic event happens after you have an adult. They in their germline, they will have meiosis to create gametes at the very end of this life cycle. And it could be considered the beginning as well. Those gametes then come together through fertilization to form the zygote. And that zygote grows through mitosis to create the diploid generation or the adult. Okay, so we're pretty familiar with that. Other types that you may or may not have kind of heard of, zygotic meiosis is actually shown on the third panel over here. This is where the meiosis takes place in the beginning. So you actually have formation of a diploid, and then this diploid undergoes meiosis to create haploid, and then you have a haploid generation that can divide through mitosis to create the gametes, which are also haploid. I have additional slides to explain each of these, but just kind of go over these briefly. For sporic or intermediate meiosis, the meiosis is not actually tied to fertilization or right at zygote formation. Okay, so this kind of happens in a different place. In sporic, you have formation of a diploid through fertilization. That diploid grows up to create the sporophyte generation, and that sporophyte generation you can see here is diploid. That undergoes meiosis to create haploid spores, but then here's the twist. Those haploid spores, instead of just fertilizing, they grow through mitosis to form what we call a gametophyte, and that's a multicellular haploid generation, and then that gametophyte through mitosis creates the gametes. So it's a little bit more complicated here. So just to kind of go through these one by one, for most multicellular animals and many protists, we see this gametic. And in the gametic, again, the meiotic divisions are closely linked, directly linked, to gamete formation. You can see down here. And then zygotic, that meiotic process is happening right after fertilization. So you form a 2N zygote, and that 2N zygote is then going to undergo meiosis to create the haploid generation. This is what we see with protists and fungi. So when we talk about Saccharomyces cerevisiae or other fungi, this is their life cycle. The third type, as I mentioned, sporic meiosis, or sometimes called intermediate meiosis. Meiotic divisions take place at stages unrelated to gamete formation or fertilization. In this case, you have diploid zygote, grows through mitosis to become a multicellular sporophyte. That sporophyte undergoes meiosis to create haploid spores. Those are 1N. And then those spores grow to form the gametophyte haploid generation. Okay, and those gametophyte then just through division, through mitotic division, can form the gametes. They don't have to undergo meiosis. So you can see here the genetic information is being shuffled at different points in these three different types of organisms. This is the sporic form of meiosis is observed in many plants and algae. Okay, so brief review of the stages. I actually hope that you can kind of review this a little bit on your own, but just like we did in genetics, our textbook goes through the stages of prophase 1. Prophase 1 is a really big, important phase for meiosis because there's a lot of things that happen that are different than mitosis. And one of the very first things that happens, well, first we do see condensation. We saw that also in mitosis. We get condensation of the chromosomes. You can see here they're beginning to condense. You have the movement of centrosomes to opposite poles, and all of this is what we do see in mitosis. So this is occurring in leptotene, which means thin and delicate. So this is still before they become really observable thick filaments. This next phase is called zygotene, and you can see it here. This is when pairing happens, and this is the first difference that we see between mitosis and meiosis, is that in meiosis, during prophase, you get pairing of homologous chromosomes. That's really important because that's going to allow them to form crossovers, and that is one of our key ways that we create genetic diversity. So this stage is called zygotene. I remember this just because a zygote is fusion of egg and sperm, and so the zygote here in this case is fusion of the two homologous pairs. Okay, so it's the conjugation stage. It's also called synapsis. So this term just means pairing of homologous chromosomes. The pairing is majorly mediated by what we call the synaptonemal complex. This is like a zipper that identifies sequences that are analogous on two homologous chromosomes. And it's these, you can see the synaptonemal complex proteins kind of form a zipper in between the two. When they come together, they are then called, and you can, it's the very tiny words right here, but it's an important term to know, they become what we call the tetrad. So let's just erase this. So a tetrad, or also it can be called a bivalent. That's another term. They mean the same thing. This is when you have homologous pairs that have paired up and formed this attachment. So we'll just use two different colors if you have, but these are homologous, same size. So this would be called a tetrad or a bivalent, this pair. Those two homologous chromosomes, they have the same genes, but they might have different alleles of those genes, right? But they're the same, the chromosomes are the same size, they have the same general content, but they might have variation in what forms of the genes are present. Okay, so at this stage, when the synaptonemal complex is forming, you begin to see what we call recombination nodules. There's proteins, we're not going to go into the details of this, but there's proteins that are recruited to these recombination nodules that you can see here, these red balls, And those proteins induce double-strand breaks in one of the chromosomes, and the homologous pair actually invades into this, and they have an exchange of information. Your textbook does actually detail it a little bit in section 14.15. I don't expect you to know the details, but if you're interested, you can look at that. That crossing over event forms a physical attachment between these two, because the synaptonemal complex won't be there the whole time. This dissolves in later prophase, but the crossover will be a link that will hold the two homologous chromosomes together in later stages. Okay, so we had zygotine, the pairing. Pacotine is when crossing over happens to hold these together. The next stage is called diplotin. Diplotin is double. And what actually happens is we do see the dissolving of the synaptonemal complex, so you can actually begin to see the crossover events. The tetrads become visible, and they're attached, as I said, through the chiasmata. So chiasmata are the locations of the crossing over. They don't get resolved until anaphase, when you actually have to pull these guys apart. They will remain there as an attachment point. Okay. In many species, I want to make a point right here because this will come into play, I think the paper we have on Monday, but in many species, there is a meiotic arrest that occurs at the stage of diplotin, when they're basically being held together by their chiasmata. And in humans, right, this happens when a baby girl is in her mother, is when she actually, all of her eggs or all of her oocytes will be arrested at this stage of diplotin in prophase 1. Okay. Just to kind of show you what these chiasmata look like, you can physically observe them with electron microscopy. This is scanning electron microscopy, and you can see the physical cross-links that have occurred along this highly condensed chromosome. And in this case, you can follow the tracings. These X's are where we have crossing over events. Now, if you remember back to genetics, this typically happens only in 1 to 2 locations. It's pretty rare to get double crossovers, and even more rare getting a third crossover. So in most cases, each pair will have 1 or 2 crossing over events that happens. Okay, so we've got our chiasmata. Next stage is called diakinesis. You know, kinesis is movement, so dia is double movement. You get further condensation here of the chromatids. This is where we see the late stage prophase events happening. We see breakdown of the nuclear envelope. We see the formation of the spindle apparatus, so we actually begin to see these microtubules emanating out at this point. Okay. And then we move into the next stage here. So we have metaphase. In metaphase 1, here is another difference. So we talked about differences in prophase. Here's another major difference. What lines up at the metaphase plate is different in mitosis and in metaphase 1 of meiosis. In metaphase 1, instead of having individual chromosomes line up, you have the tetrads line up, the homologous pairs that are lined up. And so when they're attaching, this gives you kind of an in-depth view here. What's attaching is actually the microtubules from opposite poles are from, well, if you look at this green pair right here, this chromosome has both of its sisters attached to the same pole, and this one has both of its sisters attached to the same pole. So here's the difference. In meiosis 1, we're going to separate the tetrad in the first division, and then in the second division, then we'll have the separation of the sister chromatids. So for this blank, what lines up along the metaphase plate? It's the homologous pairs, or the tetrads. I'm just going to, this right here, tetrad, is what should go in the blank. The tetrads line up along the metaphase plate, and the tetrads are what are going to separate in anaphase 1. Another point is that here, the chiasmata, which are holding these two together, still is a physical link in metaphase. That does not dissolve until we get to anaphase. So here you can see now we can see the physical exchange of chromosomes. Okay, I think several people have asked about how can you regulate this differently in mitosis and meiosis. In mitosis, you have individual sister chromatids that are both getting attached to opposite poles. And so it makes sense there's going to be tension pulling those two apart. This is different in meiosis. In meiosis, in metaphase 1, you can see these kinetochores of a sister chromatid pair are attached to the microtubules from the same pole. So there's not this tension in between them to separate them. So when we pull them apart, we actually are pulling one entire pair to one side and the other pair to the other side. These still have cohesin here, and this is partially regulated by shigoshin. Somebody was asking about this a little bit earlier. This is regulated again by shigoshin, which shigoshin, remember, means guardian spirit, and it's the protein that basically recruits in phosphatase, PP2A, and that dephosphorylates the cohesin and keeps it intact. So this plays a role also in meiosis 1. And in this case, there's no tension, and so these two stay together, and it's not until the next round that we actually have this bi-oriented tension where shigoshin will be inactivated. So shigoshin gets phosphorylated when there's this tension across, and it moves and therefore doesn't recruit phosphatase in, and so because of that, you get the cleavage of cohesin and the separation of sisters in the second round. Okay. You don't need to know the details of the shigoshin here. I don't have it on the slide, but just for those of you who were kind of wondering about that, I included that detail. Okay, on to meiosis. Well, meiosis 1, telophase. We have to reform our nuclear envelope here. Okay, so you get reformation of the nuclear envelope, and we form two cells. Now, if we look at these two cells, these cells have half the number of chromosomes that our starting cell had. So meiosis 1 reduces the number of chromosomes from 2n to 1n. So for the blanks, 2n to 1n. Okay, so that's why we call it reductional division in meiosis 1. So that's another difference, right? Mitosis does not change the number of chromosomes. Meiosis 2 we consider an equational division because we are not going to have a change in the number of chromosomes. So I just have some of the major points here. In prophase 2, we no longer have pairing. Why? There's no homologous pairs in the cells to pair up with anyhow. So you have, in this case, one long chromosome, one short chromosome. Those are not homologous pairs. They do not form tetrads. Crossing over also does not occur in this stage. Crossing over has already occurred in prophase 1. So really we have the major events. We have condensation for the condensation. We have, again, formation of the mitotic spindle, breakdown of the nuclear envelope. That's all happening here. Metaphase 2, now we look at what is lining up at the metaphase plate. It's just like what lined up in mitosis. Individual chromosomes line up. And so it's the individual chromosomes, the sister chromatids, that are going to separate during anaphase. Okay, so for this blank right here, individual chromosomes align. So individual chromosomes align at the metaphase plate during metaphase 2, and sister chromatids separate into separate cells during anaphase 2. Do we have a change in ploidy? No, because we still have one big, one small chromosome. It's a matter of whether they have their Xerox copy or they don't have their Xerox copy, but the ploidy number here is the same. Okay, final product of meiosis 2 is 4 total cells, and those cells are haploid. So the final product should be 4 haploid cells. 4 haploid cells. That's different than mitosis, right? In mitosis, we created 2 diploid cells. Okay, so hopefully through that comparison, you got some of the major differences between mitosis and meiosis. I did put up a worksheet, and on that worksheet there's several charts where it asks you to fill in the number of chromosomes, the number of chromatids in various stages of mitosis or meiosis. Just to clarify, chromosome means it's a unique set of information. If you just make an exact copy of it, that's not called a new chromosome, that's just called a chromatid. So you're asked to say the number of chromosomes and the number of chromatids in these various different stages. And then on that worksheet, it also asks you to look at the differences. Where do we see differences between mitosis and meiosis? Okay, so kind of to summarize here, just this chart should help you to kind of point out some of the differences. A lot of the differences happen during these two stages, prophase 1 and metaphase 1, right? Also, we can talk about differences in the products. What are we creating here? What are we creating here? So I want you to try to do that on your own. If you do have questions, please ask, and or during office hours, ask about those differences. Okay, how do we get the genetic diversity? We said that we create these 4 haploid daughter cells. They're genetically diverse. Where are the events that generate all of that diversity? Well, there's 2 major events during meiosis, and then there's an event just at fertilization that contributes to the genetic diversity of all of us. So the first source, actually I think number 2 really happens first. We should talk about number 2 before we talk about number 1. Crossing over occurs in prophase 1. I'm actually going to write that on the screen. So crossing over that happens during prophase 1 of meiosis is one of the first sources of genetic diversity, and that's because you're creating combinations of alleles that have never existed before. It's a combination of a maternal set of alleles and a paternal set of alleles that have never been together before, and you create something that's entirely new and has never been seen. Okay, so that's one source of diversity. The second source is what we call independent assortment, and this is kind of a review from genetics. But if you remember in metaphase, this happens in metaphase 1, when we were lining these up, lining all of the chromosomes up at the metaphase plate, there's choices, or there's actually not choices, it's random alignment of whether you have maternal chromosome on this side, paternal chromosome on this side, or the opposite. So for each chromosome pair, you can randomly align. And what that looks like, just to kind of show you visually, let's just say that you had 2 chromosomes total. I have to do black and green. These are all my dead markers. Okay. Okay, so if I just have 2, long chromosome and short chromosome, these could line up in this manner, and in that case I'm going to get all of the maternal copies on one side, all of the paternal copies on the other side, but it's equal likelihood that I actually have them flipped this way. Okay, so if we look at humans, we have 23 pairs, right? 23 pairs of chromosomes lining up. Each one of our pairs can decide which way they line up. That gives us a huge amount of diversity in what kind of gametes we can create. So this aligning part is happening in metaphase 1. Okay, and then the third contributor to genetic diversity of the zygote, of course, is just random fertilization. You have all this variety in one individual times all the variety you have in another individual. That's going to make a huge amount of genetic diversity. This just kind of gives you a drastic underestimation of the amount of variability. If we look at 23 pairs, there's 2 to the 23rd power number of possible arrangements. That's 8.4 million possible gametes. We're not even factoring in crossing over variation here. And if you just simply multiply 8.4 million times 8.4 million, we have 70 trillion possibilities. So there's a huge amount of diversity that you can get, and that's probably why my two boys look so different from each other. They don't really look related. Okay, so let's look at the process then of gametogenesis. How do we create those gametes? This process occurs in the germline. In males, this is happening in the testes. In females, this is happening in the ovaries. And the process itself will vary in different species. But we're going to look at the human gametogenesis process. In humans, spermatogenesis will produce 4 sperm for each primary spermatocyte. Whereas oogenesis, we will create a single ovum or egg for each primary oocyte, which means that we're going to stockpile everything into one of those cells. Even though we had potential to make 4 cells, because the egg is going to supply all of the RNAs, proteins, and organelles, most of the organelles, not all, for the zygote, we've got to kind of stockpile everything into one of those cells, and the other cells will be what we call polar bodies, and they will just be dissolved. Okay, so let's start with the simplest form first, and I'm going to my notes here. For spermatogenesis, obviously the location for this is in the testes. These germ cells are called spermatogonia, and you can see that these spermatogonia exist in the testes. Those are will give rise to the gametes. Through the process of mitosis, you create the primary spermatocyte. And this primary spermatocyte will actually undergo meiosis 1 to create 2 secondary spermatocytes. And those secondary spermatocytes will then undergo meiosis 2 to create what we call spermatids. Now, obviously, a spermatid is simply the DNA. Very little cytoplasm in these cells because obviously the sperm's not going to provide a whole lot in the way of cytoplasm or organelles. But the spermatid has to be developed through the process of differentiation. And that, of course, forms this flagellum that is needed for the motility of the sperm. It also packages a number of different things into the head of the sperm, which will be needed when this sperm has to break through and fertilize an egg. So there's a number of proteases that are held within this compartment and other factors that are used for cell-cell recognition, binding, and then breaking through for the fertilization step. You can see that in the testes, these are actually, in addition to being organized kind of sequentially, they are the actual organization of those cells kind of follows what's happening chronologically. So if you look in the outer region here, you see the spermatogonia. The next layer of cells are primary spermatocytes. The next layer is the secondary spermatocytes. And then you have the spermatids and formation, of course, of the mature sperm. This entire process of spermatogenesis takes about 60 days, 48 to 60 days. And so each phase here occurs about every 20 days. The amazing thing for spermatogenesis is that it is occurring throughout the life of a male. This is why you can have males that are in their 70s that are fathers, that they are continuously producing sperm after puberty pretty much for the rest of their life. Now, testosterone, which regulates this process, does decline in a man's life, so they may not produce as many sperm as they once did, but they are producing sperm throughout their life. So this is in contrast, of course, to oogenesis. Many differences here. Oogenesis is occurring, is actually, a lot of this actually occurs before a woman is even born. It occurs in the fetal ovary. And so you have basically, in the fetal ovary, you have these oogonia, which divide through mitosis, just like we had the spermatogonia. But they divide through mitosis and then through to form the primary oocyte. Okay, that primary oocyte begins the process of meiosis 1. However, it arrests in diplotin stage of prophase 1. So it's going to arrest early on in this process, and there it sits for years. And it sits basically, arrests at diplotin phase of meiosis 1 until ovulation. So each month, one or a couple of oocytes will re-enter meiosis and will complete meiosis 1 here, and will actually move into meiosis 2, but they don't complete meiosis 2 unless fertilized. So there's a second arrest each month. The oocytes that have begun this process, they will arrest at metaphase 2, and if they don't get fertilized, they will just be sloughed off, right? If they get fertilized, they'll complete meiosis 2. Actually, after the sperm DNA has entered the egg, they'll complete meiosis, and then you'll see the fusion of the pronucleus from the female and the pronucleus from the male. Okay, so this process is incredibly different, right? There's all these starts and stops, and if, depending on how long into your life cycle you are, if you're in your 40s, right, and you're still ovulating, those eggs that you have there, those oocytes, I should say, have been arrested for 40 years. And they've been arrested in, right after they crossed over, and they have to resolve those crossing over events, re-enter meiosis. And as you know, as we age, our machinery isn't quite as proficient at doing what it's supposed to do, and so there can be more problems as we age in completing this process. And that's the reason why we tend to see a link between the mother's age and the incidence of some of these chromosomal abnormalities. I will say, however, that in recent studies, they are looking and identifying some things that are going wrong on the male side as well. But for things like Down syndrome or other chromosomal aneuploidies, they are most related to the female's age. Okay, so that is basically the sort of compare and contrast spermatogenesis and oogenesis. The one last slide that I included, just because next Monday in our presentation, the group is going to be talking about what happens when meiosis goes awry. One of the major events that can happen is non-disjunction, and that's just what it sounds like. Not disjoining means that sister chromatids stay together in some cases. This can happen in either the first meiosis or the second meiosis, can occur at either stage, but either way, it's going to give rise to gametes that are either have an extra chromatid or have an extra chromosome, or they're missing a chromosome. And so this is going to result in what we call aneuploidy, which I think we talked about that word. We talked about that in genetics a bit. This can give rise to a number of different developmental disorders. Down syndrome, we talked about Turner syndrome and Klinefelter syndrome, where you have non-disjunction of the sex chromosomes. And there are other chromosomal abnormalities that can occur. Most of the others are actually fatal. So there's trisomy 13, trisomy 18. Those are both fatal events where the baby might make it to birth, but they don't survive beyond birth. Okay, so that's just sort of a preview for our final presentation on Monday. If you can, try to finish the worksheet, and if you have any questions, we can talk about those questions on Monday or Tuesday. So yes, that I believe is it. Hopefully it wasn't too long. Let me see.